All right, so let's talk about epistasis. This is kind of an interesting one where we have one gene that's going to affect the phenotypic expression of another gene. And I have the example over here of rats. So we're looking at a black rat, and we're going to cross this black rat with another one, and we're going to look at their offspring. So we can see that we have 4 alleles here. So we have B, which refers to the pigment color. So the capital B is dominant for black pigment, and the lowercase b is recessive for brown. So we have one gene for pigment color. Black is dominant to brown. You see the letter C, that they're heterozygous for this gene that we're coding with the letter C, which refers to if the pigment is in the hair follicle. So capital letter C is the dominant for the pigment will be deposited in the hair follicle, and lowercase c refers to the fact that the pigment is not going to be deposited in the hair follicle. So if we look at the outcomes from this cross, we see in this first block a rat that has the phenotype of being black. He has 2 dominant black alleles, so definitely will be black in color, but also has 2 dominant Cs, meaning the pigment is going to be deposited in the hair follicle. So it's producing the black pigment, and the black pigment goes into the hair follicle, and the rat appears black. If we look down here, of course I draw my circle right through the genotype right there. So we have a brown rat, and that's because it has the 2 recessive alleles for pigment color, and it has the 2 dominant alleles for having the pigment deposited into the hair follicle. So it's making brown pigment, and that pigment's going to be deposited in the hair follicle, right? So that seems normal, right? Everything seems like it should be working that way. So let's look at this individual here. This one has the phenotype of being white, but it's making black pigment, right? It's homozygous dominant for pigment color, so it's making lots of black pigment, but it is homozygous recessive for the pigment being deposited in the hair follicle. So it means it's not going to be deposited. So even though it's making the black pigment, none of that pigment is going into the hair follicle, and therefore the rat appears white because of the lack of pigment. This rat here is making brown pigment because it's homozygous recessive for pigment color, and it's also homozygous recessive for if the pigment's in the hair follicle, so it's not going to deposit any of that brown pigment in the hair follicle. So this is epistasis, where we have one gene, this gene 2, the pigment in the hair follicle, is affecting the expression of the other gene. So you don't see the expression of the other gene because of the gene 2 that's there. This is the same reason why you see a lot of black labs, like Labrador retrievers, the dogs. So you see a lot of them compared to chocolate labs or yellow labs, because as you can see, the black, right, if you cross 2 black labs, even though they're heterozygous here, you tend to get a lot more black labs than your chocolate or your yellow labs. Okay. All right. Now let's talk about genetic disorders. So what about in humans? If we have a genetic disorder and it's going to be passed on to the next generation. So let's look at examples that are controlled by a dominant allele and genetic disorders that are controlled by a recessive allele, and how inheritance works for these genetic disorders. It's not necessarily any different than what we've been talking about, but it's just something to kind of wrap your mind around a little bit too. So here we have a dominant controlled genetic disorder on the left. We know if an individual is homozygous dominant or heterozygous, as long as they have that dominant allele, then they're going to display the genetic disorder. Okay? So as long as that dominant allele is present, because it's dominant, right? Whatever that dominance controls, that's what's going to be expressed. And in many times in genetic disorders, it has to do with the fact that there's some mutation that has affected the instructions on how to make a particular protein. And the protein is non-functional, or it's not being produced correctly, and that's what's usually causing the genetic disorder. So you just need to remember that in a dominant controlled genetic disorder, there are two genotypes that will result in the genetic disorder. If an individual is homozygous recessive, then they'll have the so-called normal phenotype. I always put normal in quotations because none of us are normal. We all have mutations. So homozygous recessive is the normal phenotype. So an example of this is Huntington's disease. This is a disease that affects you later on in life. So a lot of times you do not know you have the genetic disorder until after you've gotten married and had kids and passed that on to your children. Because it is a dominant controlled genetic disorder, at least 50% of their children or 50% chance of passing it on to their children. Achondroplasia, which is a form of dwarfism, is also a dominant controlled genetic disorder. Other dominant controlled genetic disorders, if they're very severe, for the most part, disappear from the population because individuals a lot of times don't survive to reproduce and pass it on to the next generation. Recessive genetic disorders are controlled by the recessive allele. So if they are homozygous recessive, they'll show the genetic disorder. And once again, no protein's probably being produced, or something's gone wrong. There's some mutation, so the protein's not functioning. Therefore, if an individual is homozygous dominant, they have the so-called normal phenotype. They won't show the genetic disorder. If they are heterozygous, we say that they are a carrier for the genetic disorder. It doesn't mean they necessarily show the genetic disorder. They typically have the normal phenotype, but they can pass it on to the next generation. Okay, so they're able to kind of mask it behind the dominant allele, but it's still there and they can still pass it on to the next generation. So examples of these recessive genetic disorders are sickle cell disease and cystic fibrosis. So these are both that are recessive controlled genetic disorders. All right, let's do a practice problem now. So let's say if a woman, heterozygous for Huntington's disease, has children with a man who does not have Huntington's disease, what are the chances they will have a child with Huntington's disease? All right, so if I just draw this out here, I'm going to make my big H refer to Huntington's, right? Because we just said Huntington's is controlled by a dominant genetic disorder. I'm not going to write Huntington's all the way out. And I'm going to use the recessive or lowercase h for basically having no Huntington's. Okay, so no Huntington's, or no Hunt, as far as I can write out here. Okay, so if a woman is heterozygous, so if she's heterozygous, she has the dominant allele and the recessive allele, right? So I'm going to start making my Punnett square here. And so a woman heterozygous for Huntington's disease has children with a man who does not have Huntington's disease. So if he doesn't have Huntington's disease, what do you think his genotype is? Right? So if he does not have Huntington's, like no Huntington's is recessive, he has to have two recessive alleles or be homozygous recessive. Okay, so the woman is heterozygous for Huntington's, meaning two different alleles, It's a dominant controlled genetic disorder, so we have the dominant allele. Has children with a man who does not have Huntington's. In order to not have Huntington's, he has to have two recessive alleles. If he has the dominant allele, then he's going to have Huntington's. So what is the chance they will have a child with Huntington's? So we bring down our capital H there, bring across our lowercase h. Here we're going to bring across the lowercase h and bring down the h from above. We'll bring down this h and we'll pull the h over from here. And then we'll pull the h over and bring down our dominant allele. So what is the chance they will have a child with Huntington's? What's the phenotype of this individual? They have the dominant allele. They have Huntington's. What's the phenotype of this individual? They are homozygous recessive, two recessive alleles. They do not have Huntington's. This child is heterozygous. They have Huntington's. This one's homozygous recessive. They do not have Huntington's. So we're looking at this and we're seeing that 2 out of 4, right? So we could say 2 out of 4 will have Huntington's. We can simplify that, right, into 1/2, right? So if we simplify, put the 2 into the 4, it comes out to be 1/2. So basically 1/2 of the children have a chance of having Huntington's disease, or we can say 50% of the children would have Huntington's disease. Okay, so keep practicing these problems, and we'll move on and talk about some other inheritance patterns.